Have you ever wondered how that invisible code inside our cells actually shapes the grand, wild diversity of life on Earth? Well, you're in the right place. Welcome to this explainer on population genetics. Today, we're blending math, biology, and evolution to figure out exactly how species survive, adapt, and evolve over time.
Here's our roadmap for today. We'll start with the genetic blueprint, then look at the impossible ideal population, dive into the forces of evolutionary change, check out some real-world applications, and finally, peek into the future of genetics. Let's get right into it.
Section 1, the genetic blueprint, where we'll understand the foundation of populations. To really set the stage here, we need to zoom way out. Instead of just looking at an individual's DNA, population genetics looks at entire interbreeding groups. It's literally the study of genetic variation within those populations and the evolutionary forces that influence changes in allele and genotype frequencies over time. Basically, this is where the core principles of genetics meet the heavy-hitting math of evolution.
Now think of a gene pool as the master inventory of every single gene in a given group. It's the complete collection of all the genes and alleles present in a population. And having a larger gene pool is kind of like having a massive, well-stocked toolkit. It means much greater genetic diversity, which is absolutely fantastic for the long-term survival and resilience of any species.
So there's a really vital distinction we need to make here. First, we have allele frequency. This is the proportion of just one specific genetic unit, like a single dominant or recessive trait in the population. Then we have genotype frequency, which is the proportion of individuals carrying a specific pair of those alleles. Think of it this way. One measures the individual Lego bricks, while the other measures the paired blocks that individuals actually carry around. What's super interesting about this concept is just how incredibly simple the underlying math can be. Let's say we have a model with only two alleles. If the dominant allele A makes up 70% of the pool, and the recessive allele A makes up 30%, their sum always perfectly equals 1.0. And honestly, this basic, beautiful arithmetic is the foundation for all the complex evolutionary math we're going to talk about next.
Moving on to Section 2, the impossible ideal population, also known as the Hardy-Weinberg equilibrium.
Okay, don't let the math scare you off. This is the Hardy-Weinberg equilibrium model, and it describes a quote-unquote perfect population where genetic frequencies never ever change. In this equation, P is your dominant allele and Q is the recessive one. If you run them through this formula, as long as no evolutionary forces interfere, it perfectly predicts the genetic makeup of the very next generation. Pretty neat, right?
But here's the catch. For this genetic stillness to actually happen, a population has to somehow meet 5 virtually impossible conditions. There can be absolutely no mutations, 0 natural selection, no migration or gene flow, the population has to be massively large, and mating must be completely random. Yeah, good luck finding all of that naturally occurring in the wild.
Because these strict textbook assumptions are almost always violated in the real world, it begs the question, what exactly happens when nature breaks these rules?
That brings us to Section 3, Forces of Evolutionary Change and How Populations Actually Evolve. So let's step back into reality for a second. When populations inevitably violate that perfect mathematical equilibrium, it's because one or more major forces of chaos and change are at work shaping the gene pool. We're talking about mutation, natural selection, genetic drift, gene flow, or non-random mating. Let's break these down. First up, we have mutation. Mutations are a rare but absolutely crucial source of new genetic variation because they introduce completely new alleles into the mix. Now, this random error might turn out to be beneficial, like lactose tolerance in humans, or it could be harmful, or even completely neutral. But no matter what, it provides the essential raw material for any evolution to occur.
Then we have genetic drift, which honestly is all about luck. Sometimes random chance events occur, like a massive natural disaster that wipes out most of a population. This causes a bottleneck effect, or a founder effect if a small group breaks off. The result? Allele frequencies change drastically. The few survivors dictate the future gene pool entirely based on being in the right place at the right time, rather than survival of the fittest.
Next, consider the difference between gene flow and natural selection. Gene flow is simply the movement of alleles between populations through migration, which actually increases genetic diversity by mixing things up. Natural selection, on the other hand, is not random at all. It actively favors advantageous traits, steadily increasing the beneficial alleles that actually help a species survive and thrive in its environment.
All right, on to Section 4, real-world applications, or why population genetics actually matters.
So why does all this biological math actually matter outside of a textbook? Well, the real-world applications are huge. In medicine, we use it for disease risk prediction and pharmacogenomics, which basically means creating drugs tailored to your specific genetic makeup. In conservation biology, it helps monitor endangered species and prevent inbreeding. It's the absolute bedrock of human identification and DNA profiling in forensic science, and it's even used in agriculture to conserve genetic resources and improve our crops. It is literally everywhere.
But of course, this science isn't without its hurdles. We do face some serious limitations and challenges. For one, a lot of our models still assume those quote-unquote ideal populations we talked about, and unpredictable environmental factors can really complicate our predictions. On top of that, crunching these massive datasets requires advanced computational analysis, making exceptionally rare alleles incredibly tough to study.
Finally, Section 5, the future of genetics and expanding the scope of evolutionary biology.
We are quite literally living through a golden age of genetic discovery right now. We're seeing massive recent breakthroughs like whole genome sequencing and genome-wide association studies, or GWAS. We're analyzing ancient DNA to peer back in time, and using high-throughput sequencing technologies to read genetic codes at absolutely unprecedented speeds. And crucially, artificial intelligence is now supercharging our genetic analysis, helping us make sense of datasets that used to be just impossibly large.
These tools are actively paving the way for our immediate future. We are currently mapping global human genomic diversity to truly understand our collective gene pool. We're integrating this with multi-omics data, looking at everything from DNA to proteins, to get a complete biological picture. The ultimate endgame here, delivering precision medicine based entirely on population genetics. We're talking about healthcare completely tailored to your exact evolutionary history.
So, as these advanced models crack the incredibly complex codes of biodiversity and disease, and as AI fully merges with whole genome sequencing, we aren't just studying evolution anymore. We're building the tools to potentially direct it. This leaves us with a pretty profound question. How will AI and whole genome sequencing ultimately shape our own genetic future? Thank you so much for exploring this with me in today's Explainer. Keep questioning, keep learning, and I'll catch you next time.